Okay, so first of all, the title should be transcription and translation, so they're backwards. We always put the first process first. Alright, usually I start this chapter with a story on GMO. I know some of you are aware what GMO stands for, so that's genetically modified organisms. So these organisms have been modified genetically, meaning they have been introduced with a new gene, a gene from another species, so that these organisms will have desired traits, desired characteristics. I have an example here. So this is a comparison between two different corns. And speaking of corn, most of the snacks, food items, especially processed food at grocery stores, are actually made of corn. So usually you will see ingredients, and those ingredients are made of corn or from corn. And most of the corn in the US are actually genetically modified. So you probably are not aware of this, but you are eating, chances are, you are eating genetically modified organisms every day. So why are we genetically modify those organisms? Because they do provide a lot of advantages. When we look at agriculture, this is a normal corn, regular corn, and the one on the top is genetically modified corn. So you can see that the normal corn is very susceptible to pests, right? You can see the worms eating the corn. But the GM corn has been introduced a new trait. They can make their own biological weapon, which is called a Bt toxin. So these corn crops can make their own toxin that will kill the bugs that feed on them. So that's why if you use GM corn, usually you get a much higher yield and you use a lot less pesticides to kill the pests. So that's why GM crops have been so popular in the US. So how do we make GMOs? Now, of course, not like this, right? So this is the correct kind of overview of how we make GMOs. And then I'm using GMOs because the process of making GMOs is really tied to gene expression, which is transcription and translation that's included in this chapter. Okay, so basically, you are introducing a new gene, and this new gene will produce a new functional product. So a lot of times we'll say functional product, right? So that functional product is protein. I can't stress this enough, but I've been saying over and over that a gene has the genetic information, and we use that information to make the workers, the actual workers that make our bodies and perform different physiological functions, and those workers are proteins. Okay? Alright, so just real quick, if you're interested in GMO, if not, you can fast forward. If you are interested in GMOs, first of all, we have to scan probably hundreds and thousands of organisms and their genes to find one gene that can give us the desired protein product. Alright, I have to get my dog out of the bedroom so I can record. And then once we find a gene, and you can imagine this is a very small chance, right? So in the Bt corn case, we find a gene that can make the Bt toxin, and that gene comes from a soil bacterium. So bacterium is singular, right? So this is the scientific name for the bacterium that contains this Bt gene. So we take out this gene, we replicate, right? So we have so many copies of this gene, and then we are going to insert this gene or incorporate this gene into the corn genome. So this orange part is the corn genome. Okay, now even though this gene is not native, if you can use the word native in the corn genome, but once the corn genome acquires this gene, then this gene will be expressed and starts to make a Bt toxin, which is a protein. Okay, so basically now the corn has a new gene and they can make a new protein product, and this product is the biological weapon against pests and worms that feed on the corn. Okay, so this is a quick overview of gene expression and basically what I just talked about. Okay, now this is very important because this has one of the most important information or how do you say dogma in biology. So this is called the central dogma of life. So based on the name, you know how important this is, right? So you absolutely have to memorize this. You have to do this at all costs. Alright, now basically this describes the process of gene expression. Now we have 2 steps. Okay? So remember, the first step is transcription. So in transcription, the genetic information flows from the DNA to RNA, and specifically this is messenger RNA. So what does transcription mean? Transcription means you are copying, right? You are copying the exact words. So the genetic sequence, right? What kind of nucleotide you have, A, G, C, T. The sequence is copied exactly the same into this mRNA sequence. Alright, so that's the first step. And then the second step is called translation. So now we're going to use the genetic sequence in mRNA to make proteins. Now a lot of people say proteins, but just to be accurate, the end product for translation is actually amino acid chain, or you can call it polypeptide chain. Poly means many, right? So this chain has more than one amino acid. And then these chains may go through some modifications. They may have to be folded in the correct way, and then different chains may have, interact and then form a complete protein molecules. So after some modification, then you have the end product, functional product, which is protein. Okay, so if people ask you the more accurate way to describe gene expression is from DNA to RNA or mRNA to polypeptide chain, or you can say amino acid chain. Okay? If a protein has just one chain, then you can say, okay, protein is the end product of gene expression. But we know most proteins have multiple chains, right? Remember hemoglobin in red blood cells? It's red blood cells. So hemoglobin that actually binds to oxygen and carry oxygen. So hemoglobin has 4 chains, right? 4 chains. So it probably requires multiple genes to make this protein. So that's the central dogma of life. Now there's one thing that's not shown in this figure, which we discovered recently. It's very famous for HIV, that particular virus. So there's a group of a virus called a retrovirus. So their genetic material is RNA. So they can use their RNA to make DNA. So that information flows from RNA to DNA, right? It's the opposite direction as transcription. So that's kind of how some of the viruses work, right? If they have RNA, they can use special enzyme to make DNA. And then they insert the DNA into their host genome. So now the host cells become little virus-making machine. So that will make hundreds and hundreds copies of the virus, or specifically the genetic material, and also make protein products. And then you have so many, viral particles. Okay, so gene expression is different between prokaryotes and eukaryotes, and you need to know what the differences are. So the differences really stem from their cellular structures. So when we look at the gene expression, remember this is from DNA to RNA, mRNA to proteins. Okay. So we know where DNA is for eukaryotes. DNA is inside the nucleus. Okay. So the DNA is here inside the nucleus. But the second step, translation, from mRNA to protein, this is translation. And do you remember which cell organelle is involved in making polypeptide chains or making proteins? Ribosomes. And where can you find ribosomes? Can you find them inside the nucleus? No, ribosomes are outside the nucleus in the cytoplasm, right, for eukaryotes. Okay, so that means transcription, the first step, transcription is going to, I'm still talking about eukaryotes. Transcription is going to take place inside the nucleus, but translation is going to take place in the cytoplasm. So transcription and translation, they occur in different time, in different space for eukaryotes. These two processes cannot happen at the same location or at the same time. Now, for prokaryotes, their structure is less complex, right? So there's no nucleus. The DNA, the genetic material, is just kind of floating in the cytoplasm in the area called the nucleoid. All right. So once the mRNA is being produced, right, based on the DNA sequence, then because the ribosomes are also in the cytoplasm, these ribosomes can just attach to the mRNA that's still being generated, and those ribosomes will make the mRNA template to make proteins. So in prokaryotes, transcription and translation can take place at the same time, right? So as the mRNA is being transcribed, being produced, the ribosomes can just grab onto the part that has already been completed, and then they're going to use that sequence to make proteins. So again, for prokaryotes, these two processes are coupled. We call it coupled because they can happen in the same place, in the same time, right? Here, there's more description over here so you guys can read. All right, so here's a question. Transcription, translation are coupled in prokaryotes, eukaryotes, or both? All right, prokaryotes. All right, now we are going to look at very closely into the process of transcription. So first, again, very important, enzymes are the molecules that catalyze the process. So we're going to look at enzymes first. Now remember when we need to replicate DNA. So for DNA replication, what's the main enzyme that synthesizes DNA, the new DNA strand? You remember? DNA polymerase, right? So a lot of times you will see me just write out POL. So that stands for polymerase. So during transcription, because you are making mRNA now, so the enzyme is RNA polymerase. So for prokaryotes, single-cell organisms, no nucleus, their structure is very simple. They only have one RNA polymerase. For eukaryotes, we have to make some other RNA products, right? Remember, we have some, I think we mentioned some other structural RNAs, like transfer RNAs, ribosome RNAs, and some other RNAs. So for eukaryotes, we have 3 RNA polymerases. Okay, now I need you to remember one, and that's the one that produces messenger RNA. So that's number 2. All right. So number 1, number 3, you can just remember they make other types of RNA. All right. Now RNA polymerase reads DNA from 3' to 5'. So this is exactly the same as DNA replication, right? So remember, for DNA replication. Let's say this is a DNA strand. The DNA polymerase also reads the DNA strand from 3 to 5, right? So that it synthesizes the new DNA strand. Oh, it changes everything. Never mind, I was going to use a different color. So the new strand grows from 5 to 3, right? So this will be an example of the leading strand, right? So the leading strand goes from 3 to 5, and then the DNA polymerase can synthesize the new strand from 5 to 5' to 3'. It can synthesize this strand continuously. All right, now it's exactly the same thing for RNA polymerase. So the DNA template, hold on, let me see if this will work. Oh, it does change everything back to blue. So apparently you can't different things with different colors, so the different the word. So the DNA template also goes from 3 to 5, right? And then this is how RNA polymerase reads DNA from 3 to 5, and then the new mRNA is synthesized from 5 to 3. And again, remember these two strands, and these two strands have to be anti-parallel, right? So they have to go the opposite directions. So here, mRNA is synthesized from 5 to 3. Okay. So again, I'll ask you a very similar question to the one I asked for DNA replication. So the RNA polymerase adds new nucleotides to which end? 5' end or 3' end? The answer is 3' end, right? Because the new strand grows from 5 to 3. So all the new nucleotides, all the raw materials, will be added on the 3' end. Okay, now just some more detailed information on RNA polymerases. So again, remember number 2 is the main one that produces mRNA, and specifically it's called pre-mRNA. I'll talk about why we call it pre-mRNA. And then for 1 and 3, you can just kind of generally remember what they make, right? So they make other types of RNA, structural RNA. So that includes ribosome RNA, RNA, and transfer RNA, tRNA, and some nuclear pre-mRNA. Okay, now steps. So there are 3 steps. Initiation, so that's how you initiate transcription, and that process is actually pretty complicated, so we'll spend a little bit more time on that. And then the second step will be elongation, right? You're just growing the new strand, you're extending the new strand. And then the last one is termination. So termination, it just basically means the transcription ends there. Okay, so here there's a picture here and some description over here. Let me point out a few things. So in order for initiation to take place, for transcription to begin, you have to have RNA polymerase. And remember, for eukaryotes, the one that makes mRNA is polymerase number 2. So the RNA polymerase has to bind to a region of a gene called a promoter. Okay, this is a very, very important term. You have to remember what promoter is. And I'll talk about a little bit more about the promoter later. So for transcription to begin, the enzyme has to find and bind to the promoter region. Alright? And this will signal the DNA to unwind. Why does DNA have to unwind? Because you have 2 strands, right, being held together by the hydrogen bond. So you have to separate the 2 strands or unzip the 2 strands so that the RNA polymerase can use 1 strand as a template to make mRNA. Alright, now for elongation, like I said, this is pretty straightforward, right? You're just, the RNA polymerase just is synthesizing the new strand. So the strand grows longer and longer, and all the new nucleotides, the raw materials, will be added to the 3' end, right? So the 3' end keeps growing. So remember, for rRNA, you don't have any T, right? So no T. Instead, T is replaced by U. So if you have a DNA here, if the DNA has, let's say, A, T, C, the mRNA sequence would be U, right? So it's not going to be T anymore. And then A, and then G. Alright, termination, again, very straightforward. In DNA strand, you have a termination or stop sequence. So once the RNA polymerase gets to that stop sequence, it will stop. Will detach from the DNA. So that signifies the end of transcription. So the transcription process has a beginning stage called initiation, and I mentioned that earlier. This can be very complex. So I have an example here. This is a real gene sequence data. Okay, so I have some questions, just kind of fun to do. The figure shows the sequence of the insulin gene of which species? So we know most mammal species have insulin, right? So which species does this show? It can be intimidating when you look at the screenshot of this insulin gene sequence. Homo sapiens, right? So that's human. So this is a human insulin gene. Number 2, where is the insulin gene located? I.e., is located on which chromosome. So you're gonna have to read all the information, and you go down to the end from human genome sequence and chromosome 11, right? So for humans, insulin gene is located on chromosome 11. So how big is the insulin gene? How many base pairs or nucleotides? So when we describe the size of a gene, we can say how many nucleotides does that gene have. So we may say, oh, this gene has 253 nucleotides. We can say it that way. Or some people may use, so instead of nucleotides, they may say base pairs, and it's often abbreviated as BP, base pairs. It's really pretty much the same as nucleotides, because if you have a nucleotide here, A pairs with a G, right? C pairs with G. Let's say T, A, C, G. So this particular segment has 4 base pairing, right? It's the same as 4 nucleotides. So you can say 253 nucleotides, or you can say 253 base pairs. It's the same. Alright. Why doesn't it go away? Come on. I really like the drawing tool in Word or PowerPoint. This is PDF, it's not that great. Okay, so how big is the insulin gene? When you look at the gene sequence data, usually they will make things a little bit easier for you. So you can see here, they're numbered, right? So all the lines have numbers. So what do those numbers mean? You see the first one, 60. So that means in each row, you are going to have 60 nucleotides. So that's why this is 60, 120, 180. So at the end, this last one, the G there is 2,760. So there's a 4 more, right? So that will be 2,764. So this is how big the human insulin gene is. It's pretty amazing. Alright, now when you look at this insulin gene, it has almost 3,000 nucleotides or base pairs. So how does the RNA polymerase know where to start transcribing? That's pretty intimidating, right? This task. So this is how RNA polymerase finds a gene and start transcribing. So the polymerase can recognize the promoter region. So the promoter region is a special sequence of DNA that's located right upstream of the actual gene. So let's say this is the part of the gene that actually codes for proteins. Okay? Proteins. So you may have, I'm just making up things, A, T, G, G, C, T, or C, A, T. Okay? So let's say this, starting from here, this sequence will code for the actual functional polypeptide chain. Okay? So we're gonna say this A is number 1, plus 1. Okay, plus 1. So the T is gonna be plus 2, and G is gonna be plus 3, and so on. And when the RNA polymerase arrays the DNA sequence, it goes from left, well, arbitrarily, right? So we know it goes from this way, in this direction. So as the enzyme moves along the strand, it's really kind of like a water flowing. So think of water flow like a creek or a stream. You have upstream and you have a downstream, right? So the water flow is a one-directional. So it's also the same for how the RNA polymerase moves along the DNA chain. So it's only one direction, one way. So just like water flow, we can call this the actual genes from the very first nucleotide. This is downstream. Okay. And then everything above that, it's going to be upstream. Right. So everything downstream has a positive number, so plus 1, 2, 3, and then everything upstream will have a negative number. So if you have a T here, that's a negative 1. You have A, that's a negative 2. C, negative 3, so on. Okay. So once you get to the 30th nucleotide, let's say this is a G, that will be negative 30. All right. So that's upstream, next downstream. So we usually say the actual gene, this is downstream, and then right upstream, this little special DNA sequence, this is the promoter. Okay, so the RNA polymerase will bind to this region, and it knows, okay, so the promoter region ends here, and then I'm going to start transcribing from this positive one, this very first nucleotide of the gene. So that's how it works. Now the promoter region not only tells RNA polymerase where to start, it also tells it which strand to transcribe. Remember, when you look at DNA, there's two strands, right? So which strand does the enzyme transcribe? Now both strands are possible. So the promoter region will really tell the enzyme which strand you need to transcribe. Okay. So over here, this picture actually also shows some transcription factors. So usually we use TF to represent transcription factors. So the transcription factors recognize the promoter region, and then these transcription factors will recruit the RNA polymerase to bind to the promoter region. And once that binding takes place, then RNA polymerase is going to start the transcription from the positive one, the plus one nucleotide. So I mentioned the transcription factors a little bit more later. Okay, here we're gonna look at the difference in the promoter region between prokaryotes and eukaryotes. So for prokaryotes, the promoter region is pretty consistent between different organisms. Now, by saying consistent, I didn't mean their sequences are exactly the same, but the sequence is conservative, meaning you may have some variations, but the majority of the nucleotides in those sequences are very, very similar or can be the same. So usually for the prokaryotic promoter region, you have something like a segment that's really rich in T's and A's in the -10 region. Remember -10, so you count 10 nucleotides from away from the plus one nucleotide. So you're going upstream, so that's -10 region. And there's also a -35 region. So both regions, especially that -10, is very rich in T's and A's. And then -35, there's also A's and T's, but this time you may have some C's and G's. Again, these two sequences can kind of vary a little bit between different prokaryotes, like between different bacteria. And over here, we call that a Tata box. This is just T's and A's, so we call it Tata box. Now, in eukaryotes, we don't have that -10, we don't have that -35 regions. So what we have is a core promoter that's also has that Tata box. It has that Tata box in the core promoter region, but it's not -10 or -5. Okay. And then you may have some other elements like a silencer or activator that can further regulate gene transcription. Alright, so the RNA polymerase actually cannot directly recognize the promoter region, so it needs a little bit help. So you can think of RNA polymerase as like a half blind, if that helps. It can't really, detect where the promoter region is. So it needs a little bit extra help, like a guide dog or a guide person. So the helper is different between prokaryotes and eukaryotes. So in prokaryotes, the promoter recognition, the helper for RNA polymerase, is the sigma factor. Okay. So you can see this is what the complex looks like. So you can see this component. Now it has different subunits, right? This subunit is the sigma factor. So this is sigma factor. So this is the different components of the RNA polymerase. So you are going to have the sigma factor that will bind to RNA polymerase and bring RNA polymerase into the promoter region, right? Which is the -10, -35 promoter region. And then once the RNA polymerase has bind to the promoter region, then the sigma factor has completed its job, right? It's going to disassociate from the RNA polymerase. So this is how promoter recognition is done in prokaryotes. In eukaryotes, it's not just one factor. it's an army of different proteins. We mentioned the transcription factors. Right? So in eukaryotes, RNA polymerase II synthesizes mRNA. So all the transcription factors associated with RNA polymerase II also has this number 2. So we have found a bunch of the protein factors that will recognize the promoter area and recruit RNA polymerase. So based on the order of discovery, we have, so you can see E here, D, we'll have E, F, H. So all those things will bind to the promoter region first, and then they will bring the RNA polymerase to the promoter region. Okay, so this is just a more detailed description. So we have an army of different protein molecules, including the basal transcription factors, enhancers, silencers, and you can kind of read what they do. So here's an analogy for initiation, just kind of help you guys remember what the job is for each component. So I don't know if you guys know the movie Indiana Jones, right? He's always looking for treasures. So you can think of RNA polymerase as Indiana Jones, and it's looking for treasure. The treasure is the gene. So in order to find the treasure, which is the gene, Indiana Jones, the RNA polymerase, needs to find the promoter region first. So the promoter region is the treasure map. So the treasure map tells the enzyme where the gene is located, right? Because the promoter region is right next to the actual gene. The promoter region is right here. Promoter. Right, so this is downstream, and this is upstream. So the promoter region is right upstream from the actual gene. But Indiana Jones cannot find the treasure map without any help. So he needs some guide, some helper to recognize and locate the map. So for prokaryotes, that helper is the sigma factor. And then for eukaryotes, it's a variety of transcription factors. Okay, so hopefully that will be helpful. This is assignment we would do if we were in a real classroom. So similarities and differences in initiation between prokaryotes and eukaryotes. So you can focus on these 3 things: locations of transcription and translation. So for prokaryotes, they both happen in the cytoplasm. For eukaryotes, transcription happens in, it's very hard to write, nucleus, and translation occurs in cytoplasm. Number of RNA polymerase, so for prokaryotes, it's 1. For eukaryotes, it's 3. Mechanism of promoter recognition, for prokaryotes, you use that sigma factor. So just 1 protein. And then for eukaryotes, you use a variety of proteins called transcription factors, and specifically that's transcription factors 2. And there's some more answers here. Pre-mRNA and mRNA. So at the end of transcription, we actually make pre-RNA. Now these pre-RNA are not mature RNA molecules yet, so they need to be processed and modified a little bit to become the final mRNA product. So here, this doesn't apply to prokaryotes, right? It's only for eukaryotes. So eukaryotic pre-mRNAs must undergo several processing steps before they can be translated. Alright, so these steps are, there's going to be a 5' cap added to the 5' end of the mRNA strand, and then there's going to be a poly A tail added on the opposite end, the 3' end. And then there are going to be some enzymes and machinery that's going to splice out the introns. We're going to look at them in more details. So the mRNA molecules are single-stranded, right? We discussed that. DNA molecules are double-stranded, mRNA molecules are single-stranded. So without that double helix, double-strand structure, mRNA can be very vulnerable to degradation. So they actually are not very stable. They don't survive it very long. So in order for mRNA to move all the way, right, out of the nucleus into the cytoplasm, and then find the ribosomes to translate. That's not a very short and easy journey, right? So we want to make sure that these mRNA molecules are stable until they kind of fulfill their tasks. So there are two things that we can do to make them more stable and also easier for the mRNA molecules to exit the nucleus and get to ribosomes. So these are the two modifications. We're going to add a methyl guanosine triphosphate, so over here, to the 5' end of mRNA. So we'll call it a 5' cap. So think of you put on a hat on your head, that's kind of like that. And then in the end, on the 3' end, we're going to add a bunch of, like a segment of a bunch of A's. So we call it poly A. Poly means many, poly A tail. So it's a series of adenine residues. So those things can make mRNA molecules more stable and also kind of help it navigate towards the ribosomes. Alright, the third one is a little bit complicated, so just bear with me. When we look at a gene, it may contain axons. So axons are coding sequence, and introns. So introns are non-coding sequence. So how do you differentiate these two? How do you memorize these two? So you can think of the E in axons as expressed. So the E is expressed. Expressed as what? Expressed as proteins, right? So the axons are coding sequence. They will code for proteins. And the introns, say introverted, it doesn't want to be expressed. So that's the interfering non-coding sequence. It does not code for any proteins. Right? So they do not code for proteins. Now you're gonna ask, isn't that the purpose for having genes? Because they code for proteins and that's the functional product. Why do we have a non-coding sequence in our genes? So I'm going to explain that in the next slide. But first, we need to go through this splicing process. So because those introns are not coding for any proteins, but they are transcribed into messenger mRNA, so they are that sequence, the non-coding sequence, is part of the mRNA. So when you look at mRNA, I can't do different colors. If you look at this mRNA, you're going to have, I'm just going to make it thicker. So this is axon. Let's say axon 1, and then this part is intron 1. This is intron, and then this segment is the coding sequence again. So that's axon 2, and then that's intron 2, and then over here you have another coding sequence. So let's say axon 3. So this is intron 2. So this is what mRNA looks like. Now, since the introns do not code for any proteins, we actually do not want them in the final mRNA product. Okay? So this is actually pre-RNA. And in the mature final mRNA product, we don't want any introns, so only axons. So in this case, let's say we want all three of the axons. So that's what we want. So that means we have to remove this segment and this segment. Okay, so that's called the splicing. So we're going to splice these two intron segments out, and then we're going to reconnect those axons. And then this will result in the final mRNA. So the machinery that we're going to use to splice the introns out, to remove the introns, they're called the spliceosomes. So those are composed of proteins and small nuclear RNAs, and they can take out the introns and then reconnect the axons. So to answer the question why we have introns, non-coding sequence in our genes, which seems not very efficient, right? But we have discovered that with introns, we can do alternative splicing. And this way, with the same gene sequence, we can actually make multiple genes, not multiple genes, but multiple gene products. So we can make, with the same sequence, we can make multiple mRNAs, and each mRNA will make a different polypeptide. So this is how it works. Let's say this is a gene, right? You have axon 1, 2, 3, 4. And then the green part is the introns. So when you make the probably should make this a little bit clearer. When you make the pre-mRNA, the introns are transcribed into the pre-mRNA. And when you do the splicing, it's not like you only have one way to do the splicing. There are multiple possibilities, so you can do it in multiple ways. So if you take out intron 1, intron 2, and intron 3 along with that exon 4, then this is what your final mRNA looks like. So you have exon 1, 2, and 3. Now you can do it another way. So you can take out intron 1, and you can, when you take out intron 2 and intron 3, you can also take out the axon in between them. That's okay, right? It's not like you can only take out introns. You can also remove the axons next to the introns. And in this possibility, or in this alternative, you have a different mRNA, right? So instead of 1, 2, 3, you have 1, 2, 4. So different sequence, they're going to make a different protein product. Okay. So you can see that with this sequence, we have a lot of combinations, right? A lot of possibilities to make a different protein product. So this is actually a pretty efficient way to utilize the limited sequence you have to make a multiple proteins. I'm gonna skip this one. You know what, let's just cover that real quick. So remember, this is the human insulin gene, right? We talk about axons, introns, and when you look at this gene, all the black letters and the orange letters, they're actually non-coding sequence, so they don't code for any proteins. So only the blue ones, that's the mature protein, and then the red ones, they would code for polypeptides. And you're gonna ask, what's the difference between blue and red? So a lot of the proteins, before they are finalized, they have the signaling protein components, where those signaling components will kind of guide the proteins into their specific cellular components, cellular organelles to be processed. And once that's achieved, once the proteins are at their destinies, those signaling components will be removed. Okay, so they're not in the final product, but they are important. They need to be transcribed. So you can see for such a big gene sequence, the part that really matters, that really codes for proteins, are just the red and blue part. So that's actually pretty amazing when you think about that. Okay, so here's another example of axons and introns. So the red is the axon, so the red is the axon, so we have how many axons? 1, 2, 3, 4. And don't worry about the blue. The blue is just they corrected some previous mistake. I use this example. This is found in a plant species. I use this example because in this species, in the genome, they found a single nucleotide axon. So this one nucleotide, A, can be used as an axon. So that's pretty amazing, right? It's not multiple nucleotides. You can just use one nucleotide as an axon. I'm gonna skip this part. It's just about my research and the DNA repair genes.